Hey everyone, welcome back with a new lecture with how to calculate measurement uncertainty for microbiological measurements. In the previous lecture, I have explained estimation of technical uncertainty. I started by microbiological measurements procedure by collecting the lab sample. At least 10 lab samples shall be collected for measurement uncertainty estimation. Then test sample will be taken from the lab sample, which shall be homogenized well. Then 2 test portions at least shall be taken for analysis to be analyzed by different analysts, or you can increase the number of test portions as I explained before. Then in this lecture I will explain artificial contamination for the initial suspension. But first you should know what is the initial suspension. Primary dilution, initial suspension, primary dilution for the test portion. First, by mixing the test portion with the appropriate quantity of diluent. So initial suspension, you will make first dilution for the test portion. And this artificial contamination or inoculation of the target organism on the sample will be on the initial suspension. So initial suspension by dilution, first dilution for the sample for the test portion by the diluent, and then sometimes you will need to make serial dilution for the sample. If you don't know how much expected concentration for the target organism in the test sample, in this case you have to make serial dilution for the sample. Serial dilution to be able to count the number of colonies in this sample, because you can only count from 200 up to 250 colonies on the plate. So you have to make serial dilution to reduce the concentration of the target organism in the sample. Serial dilution is to reduce the concentration of organism in the sample by diluting the sample many times, usually 10 times dilution, 1 to 10, 1 ml of the sample of the test portion plus 9 ml diluent. And artificial contamination of the sample, which is the main subject of this lecture. How you will make inoculation or spiking for the target organism on the initial suspension after the first dilution. You can have naturally contaminated sample by the target organism, or you can make artificial contamination if required. So if you have naturally contaminated sample by the target organism, that will be much better, because matrix uncertainty also will be included, matrix effect will be included in your calculations. But if you don't have, in this case you can use artificial contamination for the sample. And artificial contamination of the sample can be performed by the following steps. First, you can get a reference culture or pure culture containing the target organism without any other interfering organisms, only pure culture containing target organism. That you can get from American type culture colonies. And keep this powder in the media for growth until you will have a visible growth. So it should be incubated until a visible growth to have a stock from this organism. So by these 2 steps you prepared a pure culture from the target organism that will be inoculated after that to the samples. And from this culture you will transfer to a vial, specific amount, small volume, and that will be also preserved, and other amount in the culture will be preserved well, because it will be used many times after that. And from this vial will be easy to get or to take the amount that will be spiked to the samples. But you don't know in this volume how much concentration of your target organism. So you need to assume it's 10 times to 10 up to 8 CFU per ml. You will assume only, because you don't know how much concentration of the organism in this volume. So you should, to know the concentration, the exact concentration, you should make serial dilution for this volume to know the exact concentration, and that will be 10 times dilution, 1 ml culture plus 9 ml from diluent it will be 10 up to 7. Then 1 ml culture, 9 ml diluent, 10 up to 6. Then to reach to 10 up to 8, up to 2, it will be 200 colonies CFU. So in this case you will count the colonies. So after serial dilution and you reach to 200 colonies, or as you assume, but you cannot go for 1000 colonies because you cannot count this, assume after you count the colonies, you should count the colonies, you found instead of 200 colonies, you found only 50 CFU per gram or ml. So inoculate, you need to inoculate sterile food samples, initial suspension for each sample, with the known quantity of the prepared microorganism. Then you should mix well to ensure that inoculation be strong in the sample, strong with the sample, strong mixed with the sample. So you need to spike a specific volume, and this volume should be determined before to all replicates. And as I explained before, that this spiking will be on the initial suspension after first dilution of the test portion. But how you can determine the volume that will be spiked, the specific volume that will be spiked on the initial suspensions? Example, if you need to spike 100 CFU per gram in the sample, so you need to add 2 ml from the spiking solution prepared, because you already counted the spiking solution and you found that it has 50, after serial dilutions, 50 colonies. So 100 divided by 50 equal to 2 ml. So 2 ml of spiking solution should be added to each replicate, each sample, each initial suspension, to get at the end 100 CFU per gram. So 10 gram sample plus 90 ml diluent, plus then you will add 2 ml spiking solution to get 100 CFU per gram. And the results also should be within the acceptable range. Here, if you multiply, divided by 2 or multiply to 2, 50 up to 200 CFU per gram, that will be acceptable, your acceptable range. That was the end of our lecture for today. Thank you and see you in the next lecture.